All right, we'll go ahead and get started. I'll make you guys co-host so you won't have a problem sharing your slides. All right, well, welcome everyone to the VICC Virtual Molecular Tumor Board session for today. We have a really exciting topic today that is honestly near and dear to my heart, which is structural variant detection. And so today we have Drs. Reger Mikaeel and Dr. John Thompson from NABCIS, which is an electronic genome mapping technique used to kind of optimize the detection of structural variants in the genome. So thank you so much for being with us today, both of you. This is really a treat. So thank you so much, and take it away.
Okay, Reger, why don't you introduce yourself first?
Okay. So my name is Reger Mikaeel. I am an application scientist here at NABCIS. I'm also a research associate at Yale School of Medicine. And I would like to thank you all for joining us today. And thank you so much, Beth, for giving us this opportunity to talk about the electronic genome mapping. Thank you.
Okay, that's great. Thanks for that. And thank you, Beth, for allowing us to present today because, as you said, we certainly think this is a very exciting technology with lots of applications for cancer and structural variants. I am the principal application scientist at NABCIS, so I work with both the R&D folks as well as commercial folks to try and optimize the protocols for the various specialized needs for customers, including cancer. And historically, I've been involved in DNA diagnostics, both rare disease and cancer, and also other instrumentation companies and another company, Pfizer, which you've probably also heard of. So a broad background, but today I'd like to focus on the electronic genome mapping and how we do it. And many of you may be familiar with bio-nanogenomics and optical genome mapping. Many of the practices and outputs are similar, but there are some important differences between electronic and optical mapping, and I'll go through those. Let me just share. Okay. Laser pointer. Okay, so NABCIS is based in Providence, Rhode Island, and we are now a Hitachi Group company, so they own half the company, and that's very important for us because, as I'm sure you're all aware, funding is a big issue these days, both for corporate as well as academic laboratories, and having that financial backbone to help us move things forward is a very important aspect of what we're doing. The electronic mapping is patented, and NABCIS is really the only one doing it. And although we are a Hitachi Group company, we are fully independent and working. We work with them, but we're providing the research and development as well as manufacturing at the moment. And the omics platform, which is the instrument that you can see right here, this is where the measurements are made using a detector, which I'll explain what that detector is, some reagent cartridges, and then a computer. So it's a very small footprint. It's about the size of the old standalone computers. And so you can actually put a bunch of them next to each other if you want more than one. And it's called the Omics Platform, and we had an early access program last year. We're now commercializing it and installing quite a few instruments around the country, around the US right now, and Canada and Japan. Right now, we're not available in Europe just due to resource constraints because there's only so much area we can handle, but that'll be coming soon as well as the rest of the world after Europe. So the important aspects of the omics are higher resolution. Now, it is a mapping technology, so you don't get sequence data. So it's mapping data at the resolution of about 300 base pairs. and then you can go up as high as feasible into the megabase region. The sample prep is fast. You can do it in one day and then run the sample overnight. And it's lower cost because it's essentially a very fancy voltmeter. So there are no lasers as you require with optical genome mapping, no imaging systems. So no laser can break and no imaging system can break. So it's much more robust because the only moving parts is a pump that pumps at microliters per minute, so that it's not a tremendous flow, so that it's actually quite reliable in terms of not going down for service. So I'll go through the protocol first in terms of what you do to get the instrument, and then tell you how that data is used and what you get out of it. So the critical thing is you have to have high molecular weight DNA. So standard DNA is not good enough because if you want to measure these very long structural variants, you need very long DNA. So we use the New England Biolabs Monarch kit, and it's a very reliable kit. And depending on what you start with, it can take an hour or 2 hours or so to make the DNA, and that DNA is stable essentially forever if you store it in the fridge. And then you take that DNA part of the prep from about 1 million cells and do sample prep, and that's all provided in the reagent kit that we provide. You then load it on the instrument, run it overnight, and you detect tagged molecules. And then you can go through one of two pathways. You can take all the molecules that you look at, and the size range we're looking for are typically in the order of 65 kb up to about 500 kb, with most of them between 100 and 200 kb. But again, you do get longer molecules up to 500,000 bases, and those can be critical for getting the longer variants. The molecules are then either, if you're trying to assemble the whole genome, you take them, you map them against each other. This is a reference-free alignment so that you align all the molecules and get contigs, and then those contigs are applied to the genome that you want. You can use any reference genome you want, again, because the assembly is reference-free. Most people right now use HT38, but you can also use T2T or 19 or whatever you like. And that will give you novel structural variants of sizes for 300 bases on up. Alternatively, if you don't need to discover new ones, but you want to verify something you've got, say you found them via microarrays or long-read sequencing or short-read sequencing, if you want to just verify them instead of doing an assembly, which is relatively computationally intensive and takes about a day, you can do verification where you just take the molecules and map them against a choice of patterns, the reference pattern or the variant pattern that you're interested in. So if you have a particular translocation or insertion, deletion that you think might be there, you just compare the reference and the variant and map all the molecules and see whether they map to which one. Again, this is the instrument right now. It's a single sample per run, and so we load it with just a pipetter and just stick it in here, and you can see the tag from the detector sticking out. This is what the detector looks like, and there are 256 nanochannels here, and the DNA is pumped through these channels, and then each channel has a voltmeter attached, and so you measure the voltage as the molecules move through. And this is all automated on the instrument. You just push a button to tell the instrument what to do, and it carries it out. Now, the advantages of electronic detection, one thing is a very low false positive rate. You don't amplify the DNA, you don't ligate the DNA, and you're looking at double-stranded DNA, so you don't have to worry about any secondary structure artifacts because it's all double-stranded. You can also easily distinguish molecules from each other. The nanochannels are much wider than, for example, the nanopores you'd see in Oxford nanopore system. The principle is the same in terms of current blockade, that we apply a voltage across the nanochannel, and if there's nothing in the nanochannel, you have a constant voltage. As soon as you stick a physical object in that channel, the current is blocked and you get a voltage change. And that's what we measure. And the DNA, double-stranded DNA, when it goes in, will cause a voltage change. And then we have tags, and the tags are put on the molecules by nicking the DNA at specific places. So we use a pair of nicking enzymes from New England Biolabs, nick the DNA, extend those nicks with modified nucleotides, and then put tags on the modified nucleotides. And the tags are about the same size as the DNA. So as soon as you see a tag, you get another voltage signal, and then it will go back down to baseline. And what you do is you measure the distances between those tags in time, and then convert the time into distances. And the tags actually vary in shape at the beginning of the molecules and the end of the molecules, because as the molecule enters the nanochannel, because it's dragging a very long molecule behind it, they are slow, so the tags are wide. And as the molecule goes through, it speeds up. And so that eliminates some of the issues with overlapping tags. So if you have two molecules going through at once, you can distinguish them. And this is a mock-up of what the detector looks like. You have two inlet ports and two outlet ports. And then in the middle where you have the nanochannel, this is where you detect the voltage change. And normally we don't use any optical methods. The one exception is if we want to show you what the DNA looks like as it goes through the channel. So this DNA has been optically labeled, fluorescently labeled, so you can see it going through, and you're going to see the DNA moving from the upper wider channel through this breakpoint right here, which is where the detection is made, out through here. And so this is real-time DNA going through, and you can see there's a very long one about to come through. You see it's going very slowly in the beginning, and then as the molecule approaches the nanochannel, it starts to speed up, and then speeds up tremendously as more of the molecule goes through. And so that was about a 400 kb molecule there. And as you can see, there are times you have multiple molecules going through, so it's important to be able to distinguish those from each other. So again, we detect length or sequence-specific tag location differences relative to the reference. Because the intervals between tags are continuously variable, you only need about 6 or 7 interval distances to generate a unique alignment. Obviously, if you have repetitive sequence, it won't be unique, but for any non-repetitive sequence, this is all you need to uniquely assign to the genome. And with our tagging methods, you have an interval distance of about 4 kb between tags, so some are much smaller and some are much larger. Now, the deletions or insertions are pinpointed within an interval, but the actual resolution depends on that interval size. So if you have an interval of 4 kb, the deletion occurs in a range of plus or minus 2 kb. If it's in a smaller interval, say 1,000 bases, you're within 500 bases. Whereas if it's a 20 kb interval, well, you're off by up to 10 kb on either side. So, the average resolution is, in terms of location, is on the order of a couple kb. The resolution on distance is much smaller, so that you will know that there is say a 500 base pair insertion or a 500 base pair deletion within this, and know the size of the structural variant much more precisely. It'll be within a few hundred bases, as opposed to the resolution of exactly where it is. So there's some uncertainty here, but it provides, a much broader range of measurements than you can get with any other technology. All right, so this is what the output looks like. This is an actual molecule. This is the baseline right here, voltage. You can see it better on here. There's a little bit more of it. So you can see a constant voltage. The DNA goes through. You see a voltage change. When there's a tag, you see a spike. And then you have the time measure all these time differences. And again, because the molecules speed up, this is actually the FSHD back, which, if you're familiar with it at all, it's a series of 3,300 base pair repeats. And it just so happens each of those repeats has a single nick site in it. So each of these distances is 3,300 bases. And you can see they get closer and closer, and the tags get skinnier and skinnier as they go toward the end. And again, then you go back down to the basics. This one up here is a centromeric region, over 200 kb. And again, you can see that there's a highly repetitive sequence here, and as many centromeres have. Now, some of the centromeres can be identified like this. Others have no tag sites. They have highly repetitive sequence where you don't see any tag sites at all. And again, one of the advantages of this technology is it doesn't matter how you make the nick sites. So if you have a centromere or some other region that is devoid of tags or doesn't have enough tag density to give you high resolution, well, you can just put in a nick site where you want using CRISPR, using the nicking CRISPR. And we have shown this to work. Just put in the sgRNA of interest and then just nick it wherever you want. And now you have a new nick site on top of the other ones. And in fact, if you're interested in centromeres that have no nick sites, you can design single sgRNAs that will put in 1,000 or more sites because of the repetitive nature of the DNA. So you can tell the centromeres apart. You can't always read across them. And if you have a 2 megabase centromere, you're not going to get single molecules that read across the whole thing, but you will be able to put it in context with the neighboring sequence so that you'll be able to identify, is this the centromere of chromosome 14 or 15 or whatever is of interest. So if you're looking at Robertsonian translocations or similar type things, you're able to do that with those nicases. And this is a co-translating molecule, which I'd mentioned. So you have the baseline voltage here. It goes up when there's DNA. And then in this particular case, you saw another increase and a variety of tags. And again, because of the width of the tags, you can see these 3 tags are fairly wide, belonging to the very long molecule. These tags are very narrow, so they belong to the short one. So you can then take these tags and then align it to the human genome and see that you can actually correctly predict which of these tags go with this molecule and which ones go with this. And that allows you to get deconvolve some of these co-translocations and tell what it actually is instead of having to throw away that information. So I mentioned the 2 different pathways, the variant discovery, the variant verification. And so I'll talking a lot about HG002, which is one of the main samples that we take a look at. And so the question, do you need to look at new structural variants, or you care about a particularly hypothesized? So you just generate the molecule, the same set of molecules for each, and then you either assemble into contigs or just map versus the reference genome and the alternate genome. You compare the variant discovery assembly to the reference genome. With the variant verification, you just count how many go to the reference, how many go to the alternate, and you get either genome-wide structural variant calls or verification of the one of interest. Again, the advantages of the discovery is that you get everything, but it does require higher coverage. It is more computationally expensive. And in fact, you can also verify those again using variant verification if you want, using that same data. So I'll talk first about the assembly-based method. And again, this is software that's been generated by Hitachi, the Human Chromosome Explorer technology, and show you the output we get from there. So this is an example for HE002. And what you can see is, the output gives you the chromosome, the G bands, if you're interested in those. Then there are a series of rectangles for genes, with red ones being protein coding, and there are some black ones here. Those are non-coding genes. This is the reference in terms of what tags are predicted. So based on the sequence, you can predict where tags should be. Again, you have some very long intervals as well as some very short ones. This is the assembled contig. This is contig 11,296, and this is the actual pattern. And so what you see is that in some places there are structural variants. In this case, is the missing tag, so that tells you there's something different going on here. There are other places where there are bigger differences. Then you can find all those, and here are the chromosomal coordinates. So this is around 21.8 megabases on chromosome X. There are also a number of other things you can do. You can click on the band here, and you can get the depth. You can get the individual molecules that go into it. Because there are often inversions, you can invert the map if you want. This is the map of the molecules lined up to where they map. And so that you can see these tag sites line up with the tag sites up here. And so you can look at the coverage. Does this look realistic or not? And here's a place where there is a missing tag site, but that's because these are very close. It's not unusual to be missing something when they're that tight because there'd be less than 500 bases right there. This is a coverage pattern for this. So you can, again, see it's fairly smooth across this whole region. And again, this is about 0.5 megabase that I'm showing you right now. In terms of actual useful data, this is a sample that Coriell sample that is a Fragile X repeat, and this is the FMR1 gene right here. The region that you have expansions is this interval here. So this interval is 6 kb. And what you can see is it gets a little bit bigger in the Coriell sample as you'd predict. So you see an insertion here. Oh, and this down here, this red triangle down here, you can put in different annotation tracks that you like. So this is nomad structural variance for anything that occurs at greater than 1% in the population. So you can look to see whether the constructs you're looking at are common or not. And so you can then just take this insert and compare it to what was found or what was predicted based on the Coriell data. And in fact, it is the correct size. This is another sample. This is a region of chromosome X, and this was done in association with Boston Children's Hospital. And what was done was this is the VICC gene here, which is involved in hypophosphatemia. And we confirmed that there is a 60 kb duplication here. So it comes up as an insertion, but because this banding pattern is the same as this over here, you can tell it's actually a duplication. And again, you can play with the maps here to do things differently, because right here, this left half is all aligned nicely, whereas the right half is kind of stretched out because of the insertion. You can do the opposite. You can line up the right side and then have the left hand stretched out. And you can include the nomad track below. And again, this is not a common insertion, although it is as hypophosphatemia goes, it's a common duplication. Here's some data from HG001, or so you may know it also as 12878. So there's an insertion, about 105,000 bases on chromosome X. There's also duplication next to the CYP2D6 gene, creating CYP2D7. And again, that comes up, and it's actually a heterozygous insertion here. So you see two different maps here, one with the normal DNA and one with the CYP2D7 allele. So we also can do orthogonal confirmation. So this is comparing omics data to PacBio data. This is, again, with HG002. So taking PacBio calls and aligning or mapping them to where the omics calls are, you can see that this is for all insertions and deletions greater than 5,000 bases. You can see that the size predictions are pretty much dead on. There are occasionally outliers, and there's always a reason for the outlier. It can be difficulty in putting things in the right context because of repetitive sequences, where sometimes insertions or deletions can be called multiple different ways, and you see that in this pattern. And again, for the most part, they line up very well. This is an example of a region of chromosome 8 where we have pretty much perfect alignment, where we call a 1625 base pair deletion, as do they. There's a 14,000 base pair deletion here, which they also call, and that actually is fairly common. You can see the nomad annotation there, and then there's a 6,000 base pair insertion that we also see. Now, with PacBio, they do great with deletions down to around 100,000 bases. Insertions only typically go around to 10 to 15,000 bases because the read lengths are not long enough to span larger inserts. And this is an example. So if you look at the PacBio data on their website, they had 3 technical replicates. And so we looked at some of the variants that disagree in terms of calls. So about 10% of the variants among these 3 replicates do not agree among all 3. So that means that 90% obviously do agree. So those are pretty likely real, but you have quite a few that are not. So there's a call here for 14 kb insert in chromosome 11, around 94 million. So the question is, which of these replicates is right? And the answer is probably none of them, because what we see in that region, and we have molecules that map over this whole region, is not a 14 kb insert, but a 33 kb insert. Now, we can't get sequence information, but you can look at the tagging pattern here and see that you see the same pattern of tags in this whole insert. So it's highly likely that this is a repetitive sequence. And what happened with this PacBio call is they shortened the repeat and thought that it was an insertion of only 14 kb. These correctly could tell that they couldn't see that size insertion because 33 kb is too large for PacBio to see with their read length, but you can see it easily, and you can see what is happening with the omics call. This is another very similar example where there's one of the replicates gives you a 12 kb insertion on chromosome 15. The other two don't call it. And similarly, we see now a 32 kb insert. And again, it looks like it's a repetitive sequence because you see this pattern of doublets for tag sites. So again, we don't have the sequence there, but it looks pretty clear in terms of what's going on. You can see those doublets here as well. So this repeat is just being expanded in this particular region. All right, in terms of SV Verify, again, we don't do assemblies, we do mapping. And so what you see here, this is the data from the 60 kb duplication I showed you earlier for hypophosphatemia. And so this is the reference sequence, and so the molecules are mapped here. The important thing to know about very large insertions and deletions is it matters the breakpoint. So this is an insertion, so if you're reading across the sequence, you can think of it as A, B, C, D. If you have a duplication, you have A, B, C, but instead of going to D, you have BC again. So you have a BC that's duplicated. And so you still have 3 breakpoints. The outer 2 breakpoints, CD and AB, are the same in the reference and the variant. So those breakpoints actually provide you no information. So the fact that these molecules, span the AB breakpoint or span the CD breakpoint is meaningless because it's going to happen whether it's the reference or the alternate. The only breakpoint that really matters is CB, which does not occur in the reference. And also having some reads that actually map across this so that, this is 60 kb here, 60 kb here, and you have molecules that start before, run the whole length of the 120,000 bases here and come out on the other side. So that the fact that there are many CD or CB breakpoint molecules says that there is a duplication here, and the fact that you have some that actually span all 3 breakpoints tells you even further that this is a duplication. But if you had a larger insert, say, 200 kb, we probably would not have molecules that span both or all 3 breakpoints, but you would be able to span this one and one of the others as well. If you look at the accuracy, we're still in the process of benchmarking, but looking at the false positive, false negatives, we do pretty well. We're getting better. The key thing is, what are the things that affect the accuracy? So the calling is more accurate if the structural variant size is above 1,000 bases. So if you look at the accuracy at 300 to 500, 500 to 1,000, it's less than up above 1,000. So the sweet spot is 1,000. So if you see things less than 1,000, they're probably real, but you do have to be concerned about them. And it also matters the coverage, so that as with sequencing, if you have lower coverage, the accuracy is lower. So that if you have coverage of 10 or less at the site of the variant, you only get about 2/3 correct. Whereas if you're up over 100, you have over 90% correct. And it's similar for, well, this is for the true negatives and true positives. And then the structural variant calling is also more accurate in smaller intervals. So if you look at small SV sizes in small intervals, you get good accuracy. If you look at small intervals or small SVs in larger intervals, it's not as accurate. So these are all things that we'll be trying to improve on so that we can account for some of these differences. But what will happen is right now, we don't have a good confidence score for calling variants that will be available in the near future, but using these parameters and other similar type things to try and figure out what what makes a good variant call versus not. And then with, I mentioned the size resolution. This is the interval size and predicted one. And what you can see is that once you're up above 500 bases, essentially you call all of them. Below 500 bases, it varies. It doesn't just drop off like a cliff, but around the 50% mark is around 300 bases. So that's what we call as our resolution, that half the time we can see an interval of 300 bases and half the time we can't. Okay, and to move into the cancer field, going to talk about data from the paper that appeared in Genome Biology a couple of years ago, where they did a structural variant analysis of cancer. And what they had done is use many technologies. Unfortunately, at that time, we couldn't use EGM because it was not, it did not have high enough throughput at that time to routinely do whole human genomes, unlike the current time where now we're routinely doing whole human genomes. So what they had done was to identify structural variants, tumor versus normal cells, using Illumina, PacBio, Oxford Nanopore, Hi-C, and 10x Genomics. So that gave them a list of structural variants, and then they cut that down to what they called high confidence structural variants. So all you had to do was have two platforms agree, so that there were over 1,700 structural variant calls in the high confidence set. Only 17 of them were found by all 5 technologies. So the vast majority were actually found by only 2 of these. So sometimes it was PacBio and Oxford, sometimes Illumina and Hi-C, whatever. But in any case, there were a total of 592 high confidence insertions, duplications, and deletions above 300 bases. Again, because our resolution is not good. We can occasionally see variants below that, but they are not particularly accurate. They also took these high confidence variants and attempted to validate them using bionano OGM, or optical genome mapping, microarrays, PCR, and RNA fusions. So they took all of the 592 and checked to see how many they could see in these other 4 technologies. And it's shown here where they were analyzed the 5 technologies, compared the calls, and if there was just one technology, maybe it's a structural variant. If there were 2, 3, 4, or 5, it was high confidence. And then they took these and analyzed using 4 additional technologies. Now we took the same ones and looked at them using EGM. Again, using the tumor cell line. We also looked at the normal cell line, and those are what we call verified SVs, as in terms of verified by EGM. And this is what you see for insertions and deletions, as well as the combination. And so if you have, the technology that we used here, so with EGM, we saw 103 of the 113 variant or insertions between 300 and 1,000 bases, so we get 91%. You compare that to the PacBio data of 94%, ONT of 99%, Illumina 8%. So clearly, this is a region that some technologies don't do well. Like Illumina is a short read; it cannot span these very easily. OGM is better at larger variants, so it cannot see things this small. As you move up in size, again, EGM hit 83% of them, PacBio 78%, ONT 94%, and Illumina 41%. And so, and again, the numbers for the very large ones, again, the EGM is similar to the other long read technologies. OGM sees some of them, but not all of them. If you look at deletions, again, same story, that you see, patterns that are similar between EGM and the long read technologies, although with deletions, the long read technologies do better with the smaller ones. Then once they get larger, they're much more similar. Then the question is, well, how often do these agree? So if you look at the calls for insertions where PacBio and ONT agree, that was 96 of them, or 105 of them. So out of the 105 that the 2 long read technologies agreed upon, EGM found 96 of them, did not see 9, etc. So if you get down to the whole size range, EGM found 231 and missed 27. And if you look at the ones that only PacBio saw, we didn't agree as much. Same thing with ONT, so it also makes you wonder whether those are real or maybe it's just that they're harder to detect. We don't know at this point. But there are also some things that were not actually detected by either, and some of those the EGM detected as well. And again, this insertions and deletions. And you see with OGM, again, with the smaller things, they don't do that well. And then as they get bigger, it does better. And this is an example of one of the deletions we saw. So this is normal cells where you see there are two alleles. You see an insertion, deletion. And again, this is a spread of about a megabase from 6.3 to 7.2 on the chromosome 11. And if you look at the tumor cells, you see a deletion of 82,516 bases, and as well as another, small insertion here, so that you can clearly see the difference between the cells. And there are obviously lots of them. Most of them are not that size, and you also get a readout of the genes affected by that particular deletion. So in terms of applications, functional genomics, rare disease, I showed some examples there, and obviously what's of higher interest to this group is cancer research. So you can look at these structural variants and then try to associate them with diseases. There are also applications in cell and gene therapy in terms of, is CRISPR doing nasty things to your genome? Are you maintaining the same genome structure as you expand pluripotent stem cells? Pharmacogenomics, I showed the 2D6, 2D7 insertion, but there are certainly many others, and the CYP enzymes tend to be susceptible to quite a few rearrangements, duplications, triplications. So having a mapping, a visual of how things work is important in that area as well. And then emerging technology, the sky's the limit. I mean, there are lots of things that can be envisioned that could be done here. All the data I showed you only looked at a single size tag. Different size tags with different sizes or shapes will give you different patterns electronically. So we are exploring that as well for the future. If you need to look at a couple different things simultaneously, if you have antibody labeling of methylated regions or protein binding, you could superimpose those on the genome structure. So we haven't done that yet, but it's just lack of time. With Reger and myself at this meeting, that's 2/3 of the applications group for NAPSIS, which is why we haven't done some of these things yet. But certainly there are plans to do so as we expand our group and work with customers to tune these things to their needs. And I'll close with a AI-generated view of our DNA moving through a nanochannel and open it up for any questions that people might have. And thank you for your attention.
Great. Thank you so much for that presentation. It's reminiscent of some technologies we've seen, but it also has a different flavor to it. One thing that I have noticed in some structural variant detection technologies, and it seems like EGM has the same limitations as the high molecular weight DNA. I know a lot of tumor samples that we see in our reference laboratory are processed and formalin fixed and paraffin embedded. And so what kind of measures have laboratories taken to ensure high molecular weight DNA coming out of tumor samples?
Yeah, so we like to stay away from formalin as far away as possible. Because again, the reality is if you're looking for large variants, you need large DNA, and that's a fact of life, unfortunately. And so, the optical genome mapping uses similar size DNA. Ours is a little bit smaller because we can get away with smaller pieces because we have higher resolution. But it still needs, I mean, if you're going to see insertions or deletions of tens of thousands of bases, you need reads of over 100,000, which means you need that long a DNA. Now, again, there are some, yeah, I mean, even the technology to do the short read, I mean, if you look at Hi-C, for example, although they're short reads, you have to start with long DNA. Again, there can be certain advantages there, but again, it's a very different output that you get, and it can be challenging to interpret. I don't know if you ever tried Hi-C, but it's not easy to analyze that data. And yeah, I don't think that any single technology will ever be the answer. So you're going to have to use different things, but if you want the really long variants, you're going to need really long DNA. That's
Yeah.
just the sad truth of it. So again, we haven't done much with tumor samples, but if you look at circulating free DNA, that's a no-go. Formalin fixed is no-go. But if you have tumor cells in circulation, if you have a tumor, those are the viable samples. So we have looked at some, hematological cancer cells, and those are fine. But if
Right.
you stick them in formalin, not fine anymore.
Right. Yeah, I remember having similar restrictions when we were running mate pair sequencing as well in our laboratory. And so no, it's good to understand kind of the limitations there, and also kind of be looking forward and saying, if laboratories would want to implement something, what kind of upstream changes need to happen in order to make it possible. So
Yeah,
yeah.
one of the big difficulties we have with clinical labs is obviously getting to change protocols. Well, part of the protocol is making and extracting the DNA. It has to be long. But if you start with long DNA, you can use it for any of these sequencing technologies. In fact, if you do the Oxford Nanopore ultra long methods, they use essentially the same thing that we do in terms of the Monarch kit. So, the two would be interchangeable. There are other long DNA kits that you could potentially use. Again, we found the Monarch kit to be the best, but you can use that for PacBio, Oxford Nanopore, Illumina, whatever you want afterwards. So it's not like you have to do, if you start with the Monarch, you can do whatever you want with it afterwards.
Mm-hmm. That makes sense. Thank you. Are there any questions from the audience? Okay. Otherwise, yeah. So what are some of the more, I guess, and I know that you touched a little bit about like the sizes of different abnormalities that you've been able to detect. Kind of thinking optimistically, what is the smallest that you have detected, and what has been the largest
Well,
that you
again,
feel
I
comfortable
think,
with?
we have seen some things down as low as 100 bases or even 50 bases, but they're not reliable. And certainly we're going to miss most of those. It just depends on context. If you get lucky and have a small structural variant in a small interval, you can see it. But if it's in a larger interval, you're not going to see it. So again, I think, we have good accuracy between 300 bases and 1,000 bases, and then it improves above 1,000 bases. So it's and again, there's also the SV type, and we do right now better with insertions, very well with deletions. Translocations, inversions are still not perfect, but that has more to do with the software than with the technology. So we're trying to, again, fix the technology so that often translocations will be seen as insertions, which again, if you understand, as long as you go in knowing, understanding that something may be, but again, if you have an insertion of DNA from chromosome 12 onto a sample that was chromosome 4, you can, yeah, it's a translocation. But you may have to track down the other side. But, again, the software is not yet as advanced as BioNano, but we're trying to catch up as quickly as we can. But again, it's having the financial backing of Hitachi and the ability to improve the system considerably, I think will more than makes up for some of the current limitations. Yeah.
Thanks. I see Bob has his hand raised. Go ahead, Bob.
Oh, thanks. And Dr. Thompson, that was really a great presentation. Thank you very much. I'm not so much on the lab side as I am on the clinical side. And I wonder if you or perhaps Beth could speak a little bit about at what point would you order that? Is this, for instance, a case of a rare disease and you're just sure that there's something going on in a particular region and other technologies have failed? Or maybe you can just explain a little bit about where in the diagnostic odyssey you would order such a test.
Yeah, and that's an interesting question because there's a couple of different things that enter into the answer. One is how much do things cost? Because obviously you want to minimize the cost of all these studies, and the other is how many things do you pick up with that technology? So as was mentioned, we don't pick up single nucleotide variants. So if you have an amino acid change or a termination codon introduced by a single base change, we're not going to see it. On the other hand, if you have large insertions, deletions, we will see it. So it becomes a question, I think, of over time trying to understand how often diseases are caused by small changes versus large changes. And that's not a question that can be easily answered now because people really haven't been able to look at large changes. So you know how often you see small changes, and you know that that is a significant proportion of both rare disease as well as cancer. But you also, the diagnostic yield on rare disease is, typically 50, 60%. You know you're missing things. So it could be, SNPs that you're not seeing because you're not sequencing the right things, or it could be that they're large variants that you can't see because, you're not using the right technology. So I think that depending on the disease, you will choose. Do you like the double E one? Yeah, depending on the disease, it will determine which one you use first. So if you have, heme malignancies where you often see translocations and large structural changes, you may want to do EGM first. If you have rare diseases that are, single nucleotide changes, you'll want to do sequencing first. And then if you don't find what you want, follow up with something else. So if you don't see anything using EGM on, your leukemia, well, you may do additional sequencing. So I think it will vary. And depending on the disease, you will pick which one you do first and which one you do second. But again, it also provides the orthogonal confirmation, as I was showing, for the long read technology. So if you do that first, and you're not sure about some of these variants because they're not all perfect, and that's the thing. You make a call with PacBio, Oxford Nanopore, they are usually correct, but not always. And you want to make sure when you're making a disease call, is that variant actually there? And this is a way you can, look at them all at once.
Thank you.
That brings up another question. So what is an estimated operating cost per sample of running EGM?
So if you purchase a machine, which is just under $100,000 for everything, including the instrument and the computer, the monitor, etc. So everything you need to get started, the cost per genome is about $385 at the current time. And this is without the cost savings due to high usage. So, I mean, right now, we don't have a lot of instruments out there, so we're not getting the benefits of pricing scale yet. That will happen over time. The detectors, again, they're silicon chips, and once you figure out how to make them, they're cheap. But the hard part is figuring out how to make them so that they work right. And again, right now, one of the bigger problems is it's just a single sample per run. As our throughput increases with more channels, we expect to be able to put on multiple samples rather than just one. And we'll try to automate things. So right now, I mentioned we use the Monarch kit, which is not automated. Other people are trying to automate that system. It's harder to automate things with high molecular weight DNA, but it is possible. And so eventually it will be fully automated. It is not today. So again, it's good now, and it will only get better.
Great. Thank you. Any other questions? I guess I can expand a little bit on Bob's question too. techniques like EGM and OGM are nice for capturing things that are balanced. If you're running something like a chromosomal microarray, you're going to only capture unbalanced or copy number alterations in the genome. Something like this can really help connect the dots for things that maybe don't alter a copy number state, but are a little bit larger. Yeah, I know it's been stated, like it's not going to pick up everything that an NGS assay is going to, and there's kind of that difficult area of like 50 to 100 base pairs that feels like it gets to be very hard to pick up by NGS or by other methods. This method is larger, has larger footprints in general, but not saying that you couldn't capture something smaller and then confirm it using orthogonal Sanger RT-PCR. I'm assuming that's what you would use if you came across something that was like a 70 base pair inversion or something in a gene. You would just use like a RT-PCR to confirm.
Yeah, and the problem with PCR is if you have repetitive sequences, they're really hard to design primers.
Right.
And a lot of SVs, guess what? They occur in repetitive sequences. So many of the SVs that you have, you just cannot use PCR to find them. So it's interesting you mentioned arrays. We had, we're working with a customer that had, a variant that they had found via arrays, and they knew there was a copy number change, but they were trying to figure out what was happening. Well, we ran it on EGM, and it's like, oh, it's an inverted duplication. It was obvious. It's like you took no interpretation. Now, in terms of what functional effect that has, that's the hard, for you guys, that's going to be the hard part, but at least you know what you're starting with.
Yeah. I know the exact that exact scenario where we're looking for, is it a tandem duplication or is it inverted? Because that could have repercussions on how the gene functions or doesn't function. So, yeah, exactly. Cool. Great. Are there any other questions? Otherwise, we're almost at
Okay,
time.
and I would just, say that if there are any questions, I should have put my email address in, but it's just thompson@nabsys.com. So if there are any questions, just email me and I can respond.
I also just
Great.
wanted to say, sorry, a bit before finishing on, that there is a lot of information on our website as well. So if anyone is interested to learn more about the technique and its applications, you can check on our website, nabsys.com.
Great. Thank you so much. I really appreciate your time and being with us to share your expertise today.
Okay, well,
You're
thank you very
welcome.
much for
Thank
listening.
you.
It was great having this chance.
Thank you.
Thank you.
Thank you so much.
Bye-bye.
Bye.